In 2024, a Chinese submersible reached the bottom of the Kuril-Kamchatka Trench and pointed its cameras at a section of seafloor that no human technology had ever observed. What the high-definition footage captured at 9,533 meters below the surface was something the entire field of marine biology had spent decades insisting could not exist there. Not bacteria, not scattered microorganisms clinging to survival in the mud. Fields of blood-red tubeworms, some nearly a foot long, unfurling from the sediment like flowers rooted in total darkness. Thousands of them, surrounded by dense clusters of bivalves, alive and feeding, in water so deep that the pressure alone exceeds 8 tons per square inch. When the chief scientist returned to the surface and described what the cameras had recorded, he called it a stunning defiance of the crushing darkness. The rest of the research community had a simpler reaction. That is not supposed to be there. What nobody knew at the time was that this footage was not a single anomaly in a single trench. It was the first frame of a discovery that would stretch across 2,500 kilometers of seafloor and force a complete rewrite of what science thought it understood about life on Earth. How does a thriving animal community establish itself nearly 10 kilometers below the ocean surface, in conditions that every textbook described as a biological dead zone? The submersible responsible for the footage is called Fenduje. The name translates to striver in English, and it was designed and built entirely in China for one purpose: to reach and operate at full ocean depth. In November 2020, Fenduje descended to 10,909 meters in the Challenger Deep, the lowest point of the Mariana Trench, carrying 3 researchers inside a titanium pressure sphere. That dive set a Chinese national record and placed Fenduje within 18 meters of the deepest manned dive ever completed. Over the following years, the submersible conducted dozens of dives across multiple expeditions. The mission that changed everything was the Mir project, a collaboration launched in 2021 by Shanghai Jiao Tong University and the Institute of Deep Sea Science and Engineering. Between 2021 and 2024, Fenduje completed 33 dives across the Mariana Trench, the Kuril-Kamchatka Trench, and the Western Aleutian Trench. The crew spent a combined 150 hours on the seafloor. They collected over 1,600 sediment samples, seawater filtrates, and biological specimens from depths ranging between 6,000 and 10,900 meters. The scientists aboard expected to find microbes. Bacteria adapted to extreme pressure had been documented before. Single-celled organisms surviving at Hadal depths were not controversial. What they found instead made the microbial results look like a footnote. During dive 271 in the Kuril-Kamchatka Trench, the cameras captured something that had never been documented at this depth. Dense fields of cyboglinid polychaetes, a family of tubeworms, rose from black mud at 9,533 meters. These were not scattered individuals. They were not a handful of specimens clinging to a crack in the rock. They were colonies of thousands packed together in formations dense enough to be visible from meters away. Their red plumes extended into the water column as if they were standing in a shallow water reef, and not in the most extreme environment on the planet. Clustered around the tubeworms were bivalves, clams grouped around patches of white microbial mats that resembled snow spread across the dark sediment. The bivalves were not dormant. They were actively feeding, their tissues sustained by symbiotic bacteria living inside them. Pay attention to this next detail because it explains everything that follows. These organisms were not surviving on scraps of dead matter drifting down from the sunlit surface. They were powered by an entirely different energy system. The tubeworms, the bivalves, the microbial mats beneath them, all of it was running on methane and hydrogen sulfide seeping up through cracks in the seafloor. The process is called chemosynthesis. Microbes in the sediment convert those chemicals into organic compounds, and the animals consume those compounds or host the microbes directly inside their bodies. In every ecosystem you have ever seen or studied, the energy chain starts with the sun. Plants capture sunlight. Animals eat plants. Predators eat those animals. Remove the sun from that equation, and the entire system collapses. Every food web on the surface of the Earth depends on photosynthesis as its foundation. This ecosystem does not. It runs on the planet itself. Chemical energy leaking from the Earth's crust, converted into biological fuel by microbes, is consumed by worms and clams that have never seen a photon of light in their entire evolutionary history. The sun is irrelevant to them. It always has been, and the chemical measurements confirmed it. Pore water samples from the sediment showed methane concentrations 200 times higher than seawater can physically hold at those depths. Isotopic analysis proved the methane was biogenic, meaning it was being produced by microorganisms in the sediment, not by geological heat or volcanic activity. The microbes were manufacturing the fuel. The animals were consuming it. The system was self-sustaining. If this discovery keeps you watching past midnight, subscribe. We cover exactly this kind of research every week. The biological picture went deeper than the chemosynthetic communities alone. When the MIR team analyzed the sediment and biological samples back in the lab, the numbers that came back were staggering. Across the specimens collected from Hadal depths, the researchers identified 7,564 distinct microbial species. Of those, 89.4% had never been documented by science before. Not a handful of new species. Not a few dozen that had been overlooked. Thousands of organisms so unfamiliar that they had no classification, no name, no entry in any database on Earth. And no, that number is not a misprint. The diversity at the microbial level was extraordinary enough. What the genetic analysis revealed about the larger organisms was arguably more significant. The team sequenced the genomes of 11 species of deep-sea fish collected by Fenduje across multiple expeditions, from the Mariana Trench to the Indian Ocean. They identified roughly 230 genes directly associated with surviving extreme pressure. Most of these genes were involved in DNA repair, the fundamental process that keeps cells functioning correctly. At Hadal depths, the pressure itself damages DNA continuously, and these organisms have evolved dedicated genetic machinery to fix that damage in real time, faster and more efficiently than any surface organism needs to. All 11 fish species, separated by millions of years of evolution and thousands of kilometers of ocean, shared a single convergent genetic mutation. The same mutation. Independently evolved in every lineage the researchers examined. A change in a gene called RTF1 that improves how cells convert DNA instructions into RNA that builds proteins. Under extreme pressure, this one alteration gives cells a measurable efficiency advantage in producing the molecules they need to survive. That kind of convergent evolution tells you something specific. It tells you the solution is not random. When completely unrelated species arrive at the identical genetic answer to the same environmental problem, it means the problem has a narrow solution space. There are not many ways to solve it, and life found the same answer independently over and over again. The Hadal snailfish, the deepest living vertebrate ever documented, revealed even more targeted adaptations. Genomic analysis showed significantly elevated expression of collagen and keratin proteins in its skin and muscle tissues, creating a body that is more elastic and neutrally buoyant under crushing pressure, reducing the energy cost of simply moving through the water. Its genes for visual perception and circadian rhythm have been stripped away entirely, abandoned as irrelevant in a world where light has never existed. In their place, the snailfish evolved a duplicated ferritin gene that dramatically increases its tolerance to the reactive oxygen species generated by extreme pressure. The genome has been rewritten for a world where the surface rules do not apply. This decision to look beyond the Mariana Trench seemed small at the time. It wasn't. The Kuril-Kamchatka Trench, the Aleutian Trench, and the Mariana Trench are not the same geological structure. They are separate trenches, formed by distinct tectonic processes, and they are separated by thousands of kilometers of open ocean. The expectation was that any life found at Hadal, depths in one trench, would be genetically isolated from life in other trenches. Separate populations, cut off from each other by impassable distance and crushing water pressure. The genetic data said otherwise. Hadal snailfish populations showed evidence of cross-trench migration. These fish, living at depths exceeding 6,000 meters, were not confined to single trench systems. They were traveling between them, moving across the deep ocean floor through conditions that surface biology considers lethal, connecting populations that were supposed to be permanently separated. And the chemosynthetic communities were not confined to a single site either. Fenduje documented them across a continuous stretch of 2,500 kilometers of trench floor, from 5,800 meters to 9,533 meters deep. Not isolated pockets of life clinging to one favorable crack in the rock. A connected belt of thriving ecosystems, sustained by chemical energy, distributed across two separate trench systems. The ecological structure within these communities mirrored what you find in productive shallow water environments. Microbial mats formed the base, converting chemical energy into biological matter. Tubeworms and bivalves consumed that energy directly, hosting symbiotic bacteria inside their tissues. Amphipods, the small crustaceans that had already been documented at extreme depths, moved through the communities as scavengers and grazers. Polychaete worms processed organic material in the sediment. Snailfish hunted amphipods as apex predators. Distinct trophic levels, from primary producers to top predators, functioned in total darkness under pressures that were supposed to make any single link in that chain impossible. This is not what a marginal environment looks like. This is not a few hardy survivors enduring conditions that barely allow them to exist. This is a biome, a structured, self-sustaining, chemically powered biome stretching across thousands of kilometers of seafloor that nobody had looked at before Fenduje pointed a camera downward. But this was not just about the Earth anymore. The implications reached further than the ocean floor. Europa, one of Jupiter's moons, has a liquid water ocean beneath a shell of ice. Enceladus, a moon of Saturn, vents water vapor and organic compounds into space from cracks in its frozen surface. Both worlds have liquid water in contact with a rocky sea floor, the exact conditions where chemosynthesis operates on Earth. For decades, astrobiologists speculated that chemosynthetic life might be possible on these worlds, but the argument was always theoretical. The Fenduje footage converted it from theoretical to demonstrated. Life does not need sunlight. It does not need a temperate surface. It does not need an atmosphere. Given liquid water, chemical energy from geological activity, and enough time, biology builds itself. The Mariana Trench proved it. The Kuril-Kamchatka Trench confirmed it. And the genetic evidence showed that life does not just tolerate these conditions. It optimizes for them, strips away what it does not need, duplicates the genes that matter, and converges on the same solutions across species that have never encountered each other. Every ice-covered ocean in the solar system just became a more serious candidate than it was before. When the Mir researchers analyzed the tissues of deep-sea fish and amphipods collected from depths exceeding 10,000 meters, they found persistent organic pollutants, polybrominated diphenyl ethers, and polychlorinated biphenyls, PCBs. These industrial chemicals were manufactured in factories on the surface, carried by ocean currents, and settling into sediment in the deepest crevices of the planet. They are concentrated in the muscle and liver tissues of organisms that have lived in total darkness for millions of years, organisms that have no evolutionary context for the molecules now lodged inside their cells. These communities survive geological timescales of crushing pressure, freezing temperatures, and permanent darkness. They built functioning ecosystems powered by the Earth's own chemistry, independent of the sun entirely, and we contaminated them before we even knew they were there. The timing of the Fenduje discoveries is not incidental. China, the United States, and several other nations have expressed active interest in mining the deep-sea floor for rare earth minerals, polymetallic nodules, and cobalt-rich crusts. The International Seabed Authority, the body responsible for governing deep-sea mining in international waters, has still not finalized its regulatory framework. There are no binding rules in place. The ecosystems that Fenduje discovered exist in a legal vacuum. The chemosynthetic communities depend on the integrity of the sediment and the geological faults that deliver chemical energy to the surface. Mining operations disturb both. Sediment plumes from extraction can smother organisms across hundreds of square kilometers. Fault disruption can redirect or eliminate the chemical seeps that fuel the entire food web. Destroying a chemosynthetic ecosystem is not like clear-cutting a forest that can regrow in decades. These communities develop over geological time. Once disrupted, they do not come back on any timescale relevant to human civilization. Scientists who reviewed the study were direct about the stakes. These ecosystems may exist in similar geological formations in dozens of trenches across the Pacific Rim, the Atlantic, and the Indian Ocean. Given geological similarities between Hadal trenches worldwide, chemosynthesis-based communities might be far more widespread than anyone previously anticipated. The Fenduje footage covered a fraction of the available seafloor, a fraction of a fraction, and that fraction contained thousands of species unknown to science, a self-sustaining energy system independent of the sun, animals migrating across trenches, and biological communities spanning 2,500 kilometers that no one knew existed 5 years ago. The Hadal zone, the ocean below 6,000 meters, covers approximately 45,000 square miles of seafloor across trenches distributed throughout the world's oceans. Most of it has never been visited by a camera, a submersible, or a remotely operated vehicle of any kind. The geological conditions that support chemosynthesis, tectonic faults, sediment deposits, and subduction zones are present in virtually every Hadal trench on Earth. If what Fendouje documented across 2,500 kilometers of two trenches is representative, then beneath the surface of every major ocean basin on the planet, there are ecosystems operating on an energy source that has nothing to do with the sun, populated by species that no human has ever cataloged, building communities that could be older than anything alive on land. We sent one submersible to look at a sliver of the deep. It came back with over 7,000 species new to science, a functioning biome powered by the planet's own geology, and evidence that life connects itself across distances and pressures we assumed were impassable. The real question is not what Fendouje found. It's what's living in the trenches where nobody has looked yet, and whether it will still be there by the time we finally do. If that question follows you after this video, leave your answer in the comments. If you want to be here when the next discovery surfaces, subscribe now. The next video goes somewhere even the researchers did not expect.